Hello, everybody. This is the content video for Biology, Unit 3, Section 4. And we just finished talking about how genetic information is stored and used to make proteins. And now we're going to talk a little bit about how those pieces of information get changed over time. And sometimes this causes eventually evolution, either on a micro or a macro scale. And usually this doesn't really do anything. Sometimes it might make you sick, or it might be the reason that a individual has a disease. But overall, usually mutations, especially in humans, don't end up doing a whole lot. And we'll talk about why that is. All right, so the first thing we need to talk about is gene regulation. So turning on or turning off genes. Remember, genes are just portions of the DNA sequence, right? So if we're going to talk about a code for a specific protein, we're going to talk about operons. So an operon codes for a specific protein. And an operon could appear multiple times in a DNA strand for a single protein. That way, if one of them is damaged, there is a backup. Now, turning on or off those genes is controlled by transcription factors. Transcription factors are a bunch of different, usually hormones or chemicals in the body that impact DNA polymerase. Right? Remember, if DNA polymerase can activate on the gene, on the operon, then we can make transcription and translation happen, and we get a protein. If we can't make transcription happen, though, translation can't happen. And if those two can't happen, no protein is made. Something I also want to bring up here is the idea or concept of Hox genes. And they are genes that code for different portions of the body. So as you can see in this Drosophila, which is a fly, or an ant, it's a fly. The anterior to posterior, that means the front to the back, is coded on the 3' to 5' DNA. So you can see where each individual portion is coded. And the same is true for humans and mice, which look a lot like this in embryo. That means as a fetus, where the different colors line up to different portions of the body. And these are generally used more often in development rather than in the maintenance of life. So once you are developed, you don't need to continue growing a head, right? You have a head, so you're good to go. But if you're still in an embryo stage or a fetus, depending on, what species you are, you may need to continue growing and developing a head. So you're going to be using these oranges and purples, right? But once you're already grown that head, you don't have to anymore. All right, so change, right? Mutation is the way we do this. It is a change. Any change in a DNA sequence that affects the genetic information is a mutation. That's all it is. All right, it just affects the DNA sequence itself. And these can be caused by mutagens, which are either chemical or radiation that can affect the DNA itself. It can warp it, and it can do that in a few different ways. We're going to talk about that. Now, if a mutation affects somatic cells, body cells, versus germline cells, sex cells, it can have a different impact on the individual or on their genetic line. If it affects the somatic cells, it can only affect that one individual, right? If it happens in the somatic cells in your arm, it only affects the somatic cells in your arm. That might spread, say if it's cancer, but unless it affects germline cells, it doesn't get passed down. Now, some cancers do affect germline cells and can be hereditary. So it's important that if you have a family member who has cancer to get checked for that. There are some tests that can be ran. All right, so I know this table looks a little daunting. Don't worry, it's not that bad. This is just a bunch of information all on one slide so that I don't have to hunt around for it, right? So we have mutation types over here in the far left. side of the screen. Then we have an example, and this is just a sentence, but if you notice, all of the individual words in the sentence are 3 letters, just like the codons are only 3 nucleotides long. So it works as a kind of homologue or metaphor for it. And then the table also tells us the effect on protein function. So let's start working through these. Silent point mutations are the ones that don't really affect anything, and the protein can still be made and still be functioned as normal. So a point mutation only affects a single nucleotide. So in this instance, we see that it affects this O. This U becomes an O. That's all it does. All right. So this could be a C turning into a G, or a U turning into an A, or an A turning into a T. The U's are only in RNA, remember? So it could change that, but remember from that codon chart we used, sometimes that doesn't matter and you still come out with the same amino acids in the end protein. So you can still function as normal with that protein and you're good to go. The amino acid doesn't change. Missense point mutations, however, changes one nucleotide to another, the same as silent point mutations because it's still a point mutation, but it does make a different amino acid in the end. So the word U becomes Lu. DNA has all Lu can ask for instead of U can ask for. All right, and this change in amino acid changes the protein itself. It could change the shape of the protein and how it can actually work as an enzyme, because remember, proteins are shape dependent. It could mean that it's changed completely in the sense of, like, if the word can gets changed to a stop codon with it, like a nonsense point mutation. So far we've got silent point mutations, missense point mutations, and now we're going to talk about nonsense point mutation. Nonsense point mutation means that the protein is cut short because a stop codon appears where a stop codon wasn't before. So it just cuts off. This becomes a string of amino acids instead of a functioning protein, and it doesn't work. The next 2 types we're going to talk about are frame shift, and we're going to get the idea that we're either going to the left or to the right or something like that. So insertion mutations mean that we are putting in an extra nucleotide. I say we here, but it's the mutagen that's doing it, right? You're not choosing to input an extra nucleotide into your DNA sequence. So this changes the order of those codons, right? So DNA has all U, right? It changes because we're adding an extra Y here. But if you look at it, there's still a Y, an O, and a U, and then a CANASKFOR. So it's still happening, right? It's just that those codon sequences get shifted over 1. So instead of YOU being together, it's YYO, and instead of CAN being together for can, it's UCA. And this cascades down the line, and in a actual amino acid sequence here, we might miss a stop codon, and it can keep going because it's not hitting a stop codon. This can be very, very detrimental and can cause amino acids to be made into a protein that just doesn't function, or it could turn into a completely different protein even. The other type of frame shift is just the opposite. It has the same effect, all right? So deletion mutation means we're removing one of the nucleotides. So in this case, we removed an L. And you can see everything after the deletion is altered. Again, this protein isn't going to work. It's not going to be functional. And this is going to either be a disease or a non-functioning offspring if this happens in a germ cell. Inversion mutations and translocation mutations, I can't point to them because then that little box gets in the way. All right, so stay with me here. So an inversion mutation is when two nucleotides switch. All right, so this will change a couple of the amino acids in a protein, but in general, the protein can still be made, but it usually doesn't work, or it doesn't work the same. So we've got a protein that is non-functioning. Translocation mutation is multiple pieces of the DNA fragment and switch with each other. So instead of just a single nucleotide, we've got a whole codon or whole strips of DNA fragments being switched around, which means larger addition of readable codons or removal of readable codons. It can result in a completely different protein, can make a protein that your body normally wouldn't be able to make even. So this has a large impact, right? So remember, silent point mutation, no impact. Missense, usually a little impact, but we're generally okay. Sometimes it won't be. And it continues to get worse and worse as we go down this type of mutation list. All right, so how do changes arise in genetic information? Through mutations. Mutations can be caused by mutagens. They could also be changed by random chance. Now, these mutations don't always stick around. A lot of times, the body, and I don't mean just humans, right? I mean all life form bodies, have a way to catch these genetic mistakes and remove them from the body so that they don't actually impact you. But when they do, it can be very bad. Right? If you have any questions, remember, send me an email, reach out to me. Have a good day.